Last time we ended up looking at the process of DNA replication and how there must be two different mechanisms that are used to replicate the two strands, because your replication fork is opening up with your DNA helicase enzyme, and that is going to split the two strands right there. One strand is going to be synthesized following with the DNA helicase. There's your leading strand. And then the other strand must be made in the opposite direction, going away from the DNA helicase. And so we'll take a look at the steps in this process in a little bit more detail in this little video lecture here. And if you look at your leading strand, this uses what's known as continuous synthesis here. First, an enzyme is going to come in, and it's known as primase, and effectively what it is an enzyme that is going to make a short RNA that is complementary to a short region on that DNA strand that you have there. It's going to be using your 4 RNA bases, A, C, G, and T, A, C, G, and U, that are in their triphosphate form, and they're going to form hydrogen bonds to the DNA, and then this primase enzyme is going to come along and connect them together by burning those ATP and CTP and GTP and UTP. Those all provide energy for this process when they are hydrolyzed there. And this is going to make an RNA primer that's complementary just to a short stretch of the DNA, typically around 11 or 12 bases long right there. And once that RNA is built into your DNA, now your enzyme DNA polymerase can come along and it will have a free 3' hydroxyl group on your DNA strand that it can use to start adding new bases there. Similar to what we saw with our primase enzyme, the DNA bases, again, deoxy-ATP, CTP, deoxy-GTP, and deoxy-TTP, those bases are going to line up with the complementary bases on your parent strand or template strand of your DNA, and the DNA polymerase is going to hydrolyze those bases and use that energy to start making that covalent phosphodiester bond, connecting sugar, phosphate on your new DNA strand. And when we look at this process in E. coli, the enzyme is known as DNA polymerase 3 because it was the third enzyme discovered in the cells there. The other interesting kind of thing when we look at this enzyme is that people have found it actually does have what's known as a proofreading activity. So as it goes along, it almost feels the size of your DNA, and if you've got two of the smaller bases, remember our pyrimidines and our purines, two of the smaller bases are going to leave your DNA kind of narrow there, and the enzyme can actually feel when that happens, and likewise it can feel when you've got two of your big bases in place there. So in that case your DNA would bulge out, and this enzyme feels the DNA and it's able to proofread it when it feels these problems in the size of the DNA, and it'll chop out that section of the DNA there and start over again to replace it with the correct bases that line up. And this enzyme does make a mistake about once every billion bases that are added, so it's got a really good spell checker built into it. This is our leading strand, and so here there would be an RNA primer way over there, over by the origin of replication, and then your DNA polymerase just started adding your bases to that RNA primer, and it came chugging along here, and it's following along with your DNA helicase that is making this replication fork on the replication bubble there. And so this one is continuous because your DNA polymerase just keeps chugging along, following that DNA helicase as it opens up your two DNA strands. On the opposite side, though, that's where we run into the problem. And the solution that nature has come up with is basically do continuous synthesis over and over and over and over. And this forms what's known as Okazaki fragments on the lagging strand. And this allows discontinuous synthesis to occur. And effectively what's going on there is, if you think about this ORI right here, you can have an RNA primase land there, your DNA polymerase 3 comes in, and it starts chugging along, and it's making your new DNA in the opposite direction from the way the helicase is traveling. And as the helicase travels further down there, it opens up a new stretch of DNA, and you do the same process over again. You start with your primase, making the short little RNA primer, and then your DNA polymerase enzyme can come in and just chug along there, making a small section of DNA. You move down with your DNA helicase, opens up some new DNA, primer, and DNA polymerase. And the extra steps that are added into this when you have that discontinuous synthesis is we need to get rid of that RNA primer, because now your DNA is effectively a big chunk of DNA with a little piece of your RNA. And so another enzyme is going to come in, and this one is known as DNA polymerase 1 from our E. coli cells that are out there where this was discovered. And this DNA polymerase 1 is going to feel that DNA backbone, and when it runs into the ribose sugar, it's going to be a signal for this enzyme to basically chop out that piece of RNA that's in your DNA molecule there. And then once you chop that out, you're actually going to have a free 3' hydroxyl at the end of your DNA strand that was made there. And so this DNA polymerase enzyme can then start adding those bases that line up with your template DNA. They form those complementary hydrogen bonds, and then your DNA polymerase 1 here, rather than DNA polymerase 3, it's going to do the exact same activity. Start burning the energy in those nucleotide triphosphates and adding those bases onto the end of that process. So when we look at our Okazaki fragments, it goes primase, polymerase 3, polymerase 1. And then at the end of this process here, when you get all the way down to the end right there, and if we look at this thing, there is a little gap in our DNA strand there. Your DNA polymerase 3 chopped out that RNA primer and then added base after base after base. And at the very end here, you end up with your free 3' hydroxyl. And then this side over here, that had a 5' phosphate. Well, those are not connected together. You don't have that phosphodiester bond. So there is actually a gap in the backbone where it's missing. And another enzyme is going to come along here. And this enzyme is known as DNA ligase. And that enzyme is going to make that phosphodiester bond between your 3' hydroxyl and your 5' phosphate. Polymerases cannot make this bond. This is a totally different enzyme with a totally different activity, because all it's doing is making this phosphodiester bond right there. And to do this, it's going to use ATP hydrolysis. So this is our RNA base right there is being burned to provide the energy for the DNA ligase to actually make that phosphodiester bond. And now you've completed the process. And if you think about this leading strand right here, we've got that RNA primer at the beginning, and it's going to do the same thing. DNA polymerase 3 removes it, and your DNA ligase is going to seal the gap that the DNA polymerase fills in. And if we look at this sort of clunky drawing that I made, you can see this idea of DNA replication. If it's going in both directions in what's known as bidirectional DNA replication, think about your bacteria. It's a circular chromosome. This is going to go all the way around the circle. This is going to go around the circle. And these two replication bubbles are going to run into each other on the opposite side of that circle. And if you look at this closely, what you can see is the lagging strand is going in this direction. Think about it. It's unwinding there. Your lagging strand goes in the opposite direction. But over here, this particular thing started out leading strand. There's your RNA primer, and your DNA polymerase comes chugging along, following right there. And so if you look at this process, when these two things run into each other, this is going to come chugging along here. Your DNA polymerase 3 is adding your bases to your RNA primer in green right there. This is going to run into right there. And now your DNA polymerase 1 can come in, chop out your RNA primer, and go ahead and fill in the gap, and DNA ligase will attach these two pieces of that new strand of DNA. So effectively, your lagging strand runs into the leading strand, and then those two things are connected together with your DNA ligase. And it happens on both sides. And if you sit down and draw this out, think about what's going on in terms of the directions. I always try and write my phosphate there, and on my 3' hydroxyl side, I use the arrow to indicate this is where we would add new bases. So the top strand in blue right here is going phosphate left to the 3' hydroxyl. This is where we are adding our new bases, where the arrowhead is right there, which means this strand, when it was synthesized, has to be going in the opposite or anti-parallel direction. And if you sit down and draw this out, it takes a little while to see the process, and that's why I do recommend sitting down and drawing out these molecules to see how this works. But I definitely want people to be able to tell me the names of the enzymes and the steps that are involved in the process of DNA replication. This works great when we're looking at our bacteria chromosomes that go in a circle, and when we look at our linear chromosomes in eukaryotic cells, because there your replication bubbles are just running into each other. But there's one additional problem when we look at those linear chromosomes. And that's, if you think about what's going on the very, very end of this thing right here. The red shows where it matches up with our blue template or parent DNA strand. But the problem is, how do you actually get synthesis to start here? Because remember, we need to have an RNA primer in order for this DNA strand in red to be made. Well, what happens on the end of this where the lagging strand is? And you're going to run into a problem here, and you're going to run into a problem here. At the very end, there's no way for DNA synthesis to actually prime that DNA polymerase 3 to be able to start making a copy of your parent or template DNA strands. And it really wasn't until the 1980s that some researchers at UC Berkeley actually figured out what was going on and were able to show the enzyme that is able to handle this. And this is an enzyme that is known as telomerase enzyme. And what people have found is that on the ends of your eukaryotic DNA on both sides here, there's actually a set of repeated nucleotides there. And you can see it right here, TTA, GGG, and then you've got that T, and it does this over and over and over and over on the ends of the chromosomes. And what's really interesting about the telomerase enzyme is the holoenzyme actually includes a piece of RNA built into it. And effectively what this RNA does is it acts like a primer, and it allows this enzyme to lay down that RNA primer and to make a DNA strand opposite that. So effectively what it's doing is it's adding a repeat onto the end of this thing over and over and over, and that repeat is going to bind with your RNA, and then you can actually extend this DNA strand right there. And so when we look at this process, you're going to end up with a longer and longer end to your chromosome there, and they will add, we're talking thousands of repeats will go on to the end of these DNA strands. And once you've got that repeat, now you have this single-stranded piece of DNA, and your normal process can work. Your primase can come in here, bind to the end of that longer DNA right there, and then just keep chugging along and adding those new bases onto your DNA strand. So really kind of an interesting solution to allow the cells to be able to make their full-length DNA. And what people have found actually is as your cells age, the telomeres, or the number of these repeats on the ends of your chromosomes, actually get shorter and shorter. And they found if they added an extra telomerase enzyme to some cells and bred up some fruit fly to have this extra telomerase activity, those fruit flies actually lived a longer lifespan. So the ends of your DNA chromosome might be something that is a marker of the age of cells and could actually play a role in cells dying off. On the opposite side, adding too much telomerase to this can actually produce cancer. So it's kind of a damned if you do, damned if you don't sort of thing. So hopefully you understand a little bit more about this process of DNA replication. And remember, this process has to occur every time the cell is going to divide. They have to make a copy of their chromosomes for the daughter cells to use.